Good morning again. I hope you all had a very nice evening in Brussels yesterday. Now it's time to wrap up what was discussed yesterday afternoon. So I hope and I think you are eager to hear what was going on in the breakout sessions that you were not participating in. And now we will also have the chance to interact and to ask questions, and these questions might then inspire the roundtable discussion that we will have later today, or later this morning, actually. I need to remind you what the objectives of this colloquium are. It's to identify sectors in the agri-food system that are relevant to cell culture-derived food and food ingredients, review the state of the art of relevant concepts, technologies, and derived products, and to discuss emerging safety and methodological aspects and their impact on EFSA's risk assessment approaches. And I remind you about the rules from yesterday. You will get an indication when you can get the floor. Then raise your hand, wait for the microphone, state who you are, and keep the questions or comment short and to the point. So we start with the feedback from breakouts group 1, and the plan is to have 10 minutes reporting back from each of the breakout session. So I think I will call on Arbolaos first.
Good morning, everyone. So I'm here to discuss with you the feedback from the breakout session 1, where we discussed food safety hazards associated to cell culture-derived foods of animal or plant origin. So, in absence of a legal definition, and for the purpose of this colloquium, the term cell culture-derived foods of animal or plant origin refers to food produced by the propagation of animal or plant cells assisted by tissue engineering techniques. So, in the breakout session 1, we had 5 sub-thematic areas for discussion that had to do with the use of cells towards ensuring a safe and consistent product. We discussed about bioreactors, culture media and their components, scaffolding structures, nutrition, and also toxicological and allergenicity aspects. So, regarding the use of animal or plant-derived cells towards ensuring a safe and consistent product, the group discussed that the health history of the animal and the herd could be relevant to the safety, and also they highlighted that examining approaches may differ among animal species. Then, regarding the use of immortalized cell lines or primary cells from recurring biopsies and isolations, the group discussed that these different types of cells do not necessarily lead to a final product with the same degree of consistency. Then, about the establishment and the maintenance of cell banks, the group discussed that there is no need to reinvent the wheel because there are existing guidelines, mainly from the pharmaceutical sector, which could be adapted to enhance the robustness and reproducibility of the production process of cell culture-derived foods. Then we moved to some other aspects relevant to cells, and it was pointed out that cell isolation and subsequent cell treatment towards establishing a cell line could introduce chemical hazards. However, the group pointed out that there are available methods to readily monitor their occurrence, and also they discussed that the likelihood of them being present in the final product is expected to be low. Last but not least, it was highlighted that the phenotypic and genetic stability of cells should be tested throughout the whole production process at the different steps. Then I'm moving to bioreactors, culture media, and components. So it's very important to have a thorough information on the components and materials used at each step, and that would contribute towards predicting the hazards that could be potentially present in the final product. The cell culture-derived food production may include components which are not usually present in food, for which the safety status has not been established yet. And such components could accumulate in the cells and then end up in the final product. However, certain toxicological approaches, like the threshold of toxicological concern, TTC, could be used to investigate the safety of such components. Then it was also discussed that it would be useful to investigate whether some components used in the production of these foods, for example, the growth factors, that are not expected to be present in the final product could be considered as processing aids. And last, regarding the scale of the production process, it was discussed that a product of a small or medium scale production will not necessarily be representative of what will be produced when scaling up the process. Now let's see what we discussed about scaffolding structures. So one type of scaffolds could be reusable scaffolds, and these scaffolds could introduce chemical hazards to the final product through, for example, sterilization process residues or scaffold degradation over time. And also about reusable scaffolds, there is a cross-linking material that could migrate into the final product. Then there are the scaffolds that could be intentionally present in the final product, and this could affect, in the end, its digestibility. And depending on the material used and the production process implemented to manufacture the scaffolds, different processing contaminants may occur in the scaffold and then subsequently end up in the cell culture-derived foods. In the end, it was also highlighted that scaffolds could introduce additional allergens in the cell culture-derived foods, and allergens not necessarily related to foods of animal origin. Now, in the area of nutritional information and the concept of nutritional disadvantages for these products, the group discussed that the nutritional composition of the cell culture media, as well as the subsequent use of the nutrients by the cell during the process, will determine the concentration of these nutrients in the final product, in the final harvested biomass. Antinutrients from plant-derived materials, for example, culture media, components, or scaffolds, may also end up in the final product. And then, regarding the cell culture-derived foods of animal origin, it was highlighted that they may contain substantial quality of carbohydrates deriving from scaffolds that are intentionally present in the final products. And last, the last point is about protein quality, and the group discussed that protein quality in terms of amino acid profile and also digestibility would be relevant to be tested towards investigating the safety of these products. However, it should be investigated bearing in mind that factors, for example, the degree of cell differentiation could affect the amino acid profile. Last slide is about toxicology and allergenicity aspects. So it was discussed quite thoroughly, and it was agreed that a comprehensive compositional characterization of the components and materials used and of the final product also could potentially mitigate the need for a 90-day toxicological study. Untargeted analysis, like omics of the media after harvesting the biomass, could help to understand further the toxicological profile and the toxicological properties of the production process regarding components, materials, byproducts that could be formed. But it was pointed out that the implementation of such analysis is currently a bit challenging. Then moving to allergenicity, we can have allergenicity due to new proteins produced in the product because different genes may be expressed by the cells in the culture. We can have allergenicity that is linked to some of the components used for the growth of the cells. And also, as we also noted before, allergenicity could occur also to the components, to the material of which some scaffolds are made. And it was also brought up that in silico approaches could be informative towards predicting the allergenicity of these products. And last, allergens not linked to conventional animal products may be present in these foods, but the consumer would not expect them probably in some cases. However, this issue could be mitigated at a later stage by the proper labeling. So this is in brief the discussion points, the feedback from the breakout session 1, and the overall conclusions will be presented by Wolfgang after he brings forward the discussion points of breakout session 2 jointly. So thank you a lot, and I would be happy to answer later to any clarifications you may need.
Thank you, Hermolaus. And as you heard, and as you know, breakout group number 1 and 2, we're discussing the same topic. So Wolfgang, you may come up and summarize what you discussed in the breakout group 2.
Thank you very much. Thank you also, Hermolaus. It makes it easier for me. Because we dealt with the same issues, but we thought we'd do these same topics to complement each other. We have 2 different discussion groups. You always have some different outcomes, at least in some points. Some of, I would say even many, of the points raised by Hermolaus were also mentioned by our working group, and we largely agreed, I would say. Yeah? So we addressed the same questions here. On the establishment of the cell culture, the biopsies, and establishing a consistent product. We also mentioned that biopsy is one of the options. Hermolaus mentioned the other one, which are cell lines, basically even commercially available nowadays. Biopsy is important, of course, with regards to identity and traceability. This should always be documented, obviously. And then there was a question what kind of criteria should be applied for these animals which are sourced for cells, and was a quick and I would say easy answer that the animals, if they are fit for human consumption, fit for slaughtering, they should also be fit for sampling cells for this purpose. Yeah. If there are contaminations of biopsies, such contamination is considered as quickly self-contained, because the cells just you would see in a microscope, they would quickly deter and degenerate and die. And the same will apply also later on in the cultivation process. If you have contamination of cytolytic viruses or bacteria or fungi, you would quickly see that, and that would be self-containing. Yeah? Immortalization was interesting, the discussion. We heard that immortalization is not always applied, at least not intentionally, let's say. There are different methods. We heard from industry that they would like to avoid, or they are going to avoid, at least for the moment, GM-modified cells for such type of foods because of, first of all, consumer perception in Europe, and also that such type of foods would then fall under the GMO regulation. Lentiviruses was briefly mentioned, even though I haven't heard from those which are rather close to an application that they are using this. And if you use lentiviruses as a vector, that would also make these cells GM cells, basically. Yeah. There was a question on viral residue, which we could not answer because of lack of information and competence also in the group, I would say. But we thought that was not a big issue, because it was not really used in an advanced application. Stability of the cells, I would say there was an agreement among the group that the stability of the cells per se is not relevant when it comes to safety as such, because all the genes, even if they differ in their expression, all the genes are inherent. That's part of the chromosomes, and if new proteins, or if proteins are expressed, then it's the same proteins that are expressed throughout the embryonic development. So no new proteins are expected from such cells, and therefore the safety as such of the cells is considered not to be an issue. It is an issue when it comes to consistency of the product, obviously, because once we get an applicant gets an authorization, a novel food needs to comply with this specification. So Stability is an issue also for control purposes and to have a consistent nutritional profile and composition. Yeah. Mycoplasma was thought not to be an issue because it's not a foodborne pathogen, and you would see in your cultivation on your yields. BSE was very briefly discussed. TSE. It was rather thought not to be an issue. It's extremely difficult to be propagated in vitro. There was a comment that it was found from sampling, but this animal has been inoculated artificially with high doses. An old animal, so there you may be able to isolate something, but even then you need very proper and good conditions to isolate this pathogen. So obtained from young animals, it's extremely unlikely, if not impossible, to isolate TSE, BSE pathogens. On the medium and the components used throughout the cultivation process, that was of course more I wouldn't say controversial, but more concerns were raised as compared to the cells per se, obviously, because what comes in a production process that may also end up in a final product. So the medium obviously is one of the key points of such production process and such foods and safety of such novel foods. Generally, there seems to be a trend to not to use fetal bovine serum. Generally, it should be sterile, but we also heard that even that can be contaminated with viruses. But overall, it would be also against the purpose of such foods, because if you want to replace animals for animal welfare issues and then you use bovine fetal calf serum, that's not really the intention and the philosophy of such products we heard. Antimicrobials, there was some discussion about the use of it and whether it's allowed or not. Generally, we have the same issues and the same potential risks than when antibiotics are used in food production. Some residuals may end up in a final product. And theoretically, even so, that was considered not such a, very likely was that if cultivation is done with addition of some antibiotics, you may have a kind of stable contamination at low levels, and you may breed a kind of superbug. That, of course, would require rather long-term cultivation, and not if it's quickly done, and not if it's only used for biopsies. But these are the theoretical risks related to antimicrobials. Yeah? Hormones, of course, there are different types of hormones. Insulin, recombinant insulin, usually may be used in such products. And the question was, what about residuals of? Is it allowed? At what levels? Comments were said, yeah, but insulin is also in meat found, and that could be an approach to compare the levels in insulin in meat and in the product, in a novel food. Steroid hormones, sexual steroid hormones are usually not used, we have heard, but there may be applications for glucocorticoids. Yeah. TTC, Ermo mentioned, it's indeed an approach that may be applicable to some components if it is in compliance with the EFSA opinion on TTC. Threshold for toxicological concerns components at very low quantities found in the final product. The removal of residuals, that may be difficult and in some cases even impossible. But then it must be subject of the normal way of how risk assessment is done. What is the quantity, the exposure, and what is the known profile, and if available, health-based guidance values. Yeah. One comment was on modeling on the fate of components during cultivation that may be useful in order to address what happens with different components used in the medium. That was thought rather complementary, and there was the majority of the group thought it could not replace analysis of residuals. Accumulation of metabolic products. Cells are producing metabolites, obviously. If it's too much, the cells would just die. So there is also here a kind of self-containment of the amount of metabolic products that are accepted by the cells. Yeah, was thought rather not to be of concern. But there could be an accumulation from the reuse of medium, because you want to reuse the very expensive medium. It should not be just quickly disposed. And when medium is reused, the exposure to the surfaces are extended, obviously, and that may also result into residuals coming from the surfaces, depending on whether plastics are used or other materials. We heard also there is no food grade definition, no legal food grade definition. In principle, everything can be used, provided that the novel food is safe. But additional legislation may apply. That may be the food contact material legislation, the GMO legislation, or other legislations. Yeah? So that needs to be considered, obviously, when components are used. Scaffold, that was a rather short discussion. We had, and we had not much to add. But what was mentioned was that if scaffolds are used, obviously, you need to reduce costs. It's rather of plant origin, and that raises issues with regards to potential allergenicity. Some polysaccharides may enhance immune reactions. Some people are intolerant to certain fiber, those with irritated bowel disease. But that's also, this will be a case-by-case consideration. What we generally heard and what we have not heard was very specific components. Obviously, that's something that's innovative and that was thought to be protected apparently from the discussion we had, and that's also understandable, I must admit. Polymers, there was a consideration or comment that when polymers are used, which are not eatable and not end up in a final product, they still may be subject to the requirements of food contact materials. And then the same approach, obviously, then you should use, it should be used products that have been already authorized for the use as a food contact material. There was several times comments, and also I didn't want to hide this point, and we acknowledge it. What's a pity is for industry and some hurdle and burden is that there is no pre-submission advice from EFSA with regards to use certain components or scaffolds, which obviously results into uncertainties of those innovators and industry who are producing this type of products and who are using certain components that leaves them a bit in uncertainty. Nutritional assessment, similar comments like the first group. With regards to anti-nutrients, whatever is added theoretically should be a level of concerns. Of course, it's obvious and maybe even super fluent. Stability of the product. There was one comment, what about biogenic amines from protein degeneration? I must admit we did not discuss that further. That may come from these products may be less stable with regards to proteins. That's just a question that was raised. What was not raised during the breakout session, but was put on one slide from the survey, was about what about the stability of the product when they are cooked. About what's about polycyclic aromatic hydrocarbons as compared to conventional meat. That was a comment from the survey. The rest is similar to what Ermolau said, amino acid profile, fatty acid profile. What about the digestibility of the proteins? And more related, was also thought, what about the bioavailability of micronutrients, especially with regards to iron? Vitamin B12 is vitamin B12, but iron is not always iron. Iron in muscles is hemoglobin, which is quite well absorbable, bioavailable. And what about the iron in this novel food? Because these cells normally don't produce myoglobin. Yeah? And that's an issue. And when it comes to nutritional assessment, EFSA is usually considering the comparison to conventional meat. For what type of nutrients, conventional meat is a very relevant contributor. And these were thought as the main micronutrients, vitamin B12 and iron. One comment was, we have enough proteins in Europe, we consume enough. So amino acid profile, even if it somehow differs from the conventional meat, or even if some essential amino acids are less present in this cell culture-derived foods, it should be no problem because we are actually consuming already more than enough proteins. Yeah. Toxicology, with regards to residual, also here the comparison with the conventional comparator was mentioned. Again, insulin is also present in meat at certain amounts. The breakout session group thought that toxicological tests per se for the cells, for the cultured cells, are not necessary. And with regards to the medium and the production process, all depends what comes in. Again, that may also come, may be present in the final product. 90-day study was also thought to be avoided. Yeah. To be avoided also for animal health and welfare issues. And if all the components are well characterized, that should not be needed. TCC we mentioned. Allergenicity, here there was quite a strong opinion in the group that it would not be expected that new proteins are expressed in, at least in animal derived cell cultures. The expression profile probably will differ, but what would be the meaning if some proteins are at higher amounts or less? So allergenicity was thought not to be an issue there. Even so, it should be somehow also maybe demonstrated with some data. Very interesting, omics are thought not to be really needed. But then maybe it's needed for allergenicity assessment for the protein expression profile. But generally, the usefulness of omics was thought it may be not really needed for such type of applications. Finally, plants. That was considered different from the things we said above, because plants are usually capable to produce secondary plant metabolites to deter animals or insects, or also animals, to be consumed by them. So they are producing potential toxic components, and here that may be different than from cultured animal cells. Yeah. Thank you.
Thank you, Wolfgang. Then we move over to precision fermentation. And Esther, you are the rapporteur for the breakout session 3. So you have the floor.
Thank you very much, Helle, and good morning, everyone. Indeed, my name is Estefania Noriega Fernandez, and together with my colleague Gabriela Precup, we have coordinated the breakout sessions 3 and 4 on precision fermentation. And particularly for the breakout session 3, we would like to take this opportunity to thank our chair, Kevin Verstrepen, and also all our EFSA colleagues involved in the session for the terrific support. And of course, a big thanks to all the participants at the breakout session 3 for the active contributions and very fruitful discussions. Yesterday, Bruno Gautret mentioned that there is no regulatory definition for precision fermentation. So in order to frame the discussions at the breakout sessions 3 and 4, precision fermentation was referred as to the use of engineered microbial cell factories in the production of food ingredients, which, as already mentioned, require pre-market authorization in the European Union under different regulatory frameworks. For instance, novel foods, but also food additives, flavorings, and so on. When opening the colloquium, Guillaume Desses also mentioned that EFSA holds a long experience in the risk assessment of GMM fermentation products. And indeed, there are several sectoral and cross-cutting EFSA guidance documents available in order to support the applicants in the preparation and submission of the data required for the risk assessment of these products. But regardless of their intended use and regulatory framework, the food ingredients derived from precision fermentation can also be categorized according to their nature and degree of purification. So just to confirm that the product categories considered for discussion within the breakout sessions 3 and 4 included categories 1 and 2, which are products that are free from DNA and viable cells of the GM production microorganism, and also category 3 products that contain DNA but not viable cells of the GM production stream. So based on these considerations, I will now very briefly report the main issues raised by the stakeholders participating at the breakout session 3, which, as you can see on the slide, focus on new developments on engineered microbial cell factories and further considerations for their safety assessment. As you can see, the breakout session 4 had a different focus, but this will be presented later on by my colleague, Gabriela Precup. In relation to the safety of the chassis, the participants at the breakout session 3 agreed that precision fermentation does not necessarily imply a major disruption of our current approaches for the risk assessment of GMM fermentation products. In addition, the participants at the session also suggested to apply the concept of safety by design, which prioritizes the safety over other parameters relevant for the process and product design and development. This because this approach will allow to minimize safety issues already from the beginning, from the early stages. Finally, they concluded that in addition to genomic data, phenotypic testing might also be needed in order to complete the characterization and the risk assessment of the chassis. And this is particularly relevant in the case of filamentous fungi, when the state of the art is not sufficiently developed, when, for instance, the annotation of certain species is incomplete, or in case of new species. The participants in the session also emphasized the need to gather sufficient knowledge, sufficient information about the metabolism of the host strain when introducing new metabolic pathways, because this will allow to predict possible adverse effects, for instance, through computational modeling, and also to optimize the production of the target compound. And they also concluded that in vivo mutagenesis through CRISPR-Cas or any other new genomic technique would pose a challenge for the risk assessment due to the difficulties to discriminate between the point mutations introduced by the technique and the point mutations naturally occurring. Before reporting on this discussion point, new genomic techniques and the QPS qualified presumption of safety assessment, I would like to remind you that for GMMs for which the species of the parental or recipient strain qualifies for QPS status and for which the genetic modification doesn't give rise to safety concerns, then the QPS status could be extended to the GMM production stream. So based on this consideration, the breakout session, the members concluded that the QPS concept could also be extended in case of GMMs generated by new genomic techniques. The participants consider that the hazards associated to GMMs are independent of the technique, of the methodology used for the genetic modification, and in fact, they depend on the actual genetic modification. They also suggested that potential off-target effects could be assessed later on, for instance, through toxigenicity or pathogenicity testing. Before introducing the second bullet point, I would like also to remind you that the lowest taxonomic unit for which the QPS status is granted is at a species level, for instance, for bacteria and yeast. So then the participants of the breakout session 3 were wondering whether this QPS status should be granted at a strain level rather than at a species level. And based on this consideration, they are also wondering up to which extent this genetic modification would require a safety assessment. Finally, also the participants pointed out that the introduction of intended sequences with the purpose to barcode the production stream will allow later on to monitor the traceability of the particular GM production stream once the product is placed in the market. We also discussed in our sessions about strain stability, particularly the stability of the genetic traits in engineered microbial cell factories, whether this was a parameter relevant for the risk assessment and how to assess this parameter. And the participants concluded that this, in fact, is a relevant parameter for the risk assessment of category 4 products, which contain DNA and viable cells of the GM production stream. They also suggested that this parameter could be assessed through the sequencing of the genetic modification at different production stages. However, the participants also consider that this parameter doesn't represent a safety concern for other product categories, since, well, they pointed out that mutations also occur in nature. But nevertheless, this parameter would be of relevance, a concern for food business operators in relation to the product stability and also towards robust production systems. We also have a very interesting discussion regarding further optimization of GM production strains used in the production of already authorized products, which seems to be a common practice, as the participants in the session pointed out. So they were wondering what would be the minimum modification of the GM production strain to trigger the need of a new safety assessment. We had also a controversial discussion about horizontal gene transfer. Some participants pointed out to the very low likelihood of this event to happen. Some other participants also refer to the literature, to in vivo studies on microbiota. And there was also a reference to the 2011 E. coli outbreak in Germany. I would say that the main conclusions, although there were some discrepancies, some different opinions, were that horizontal gene transfer would be a safety issue in the case of the presence of sequences of concern. So we are talking about antimicrobial resistant genes or genes coding for toxin production. And they also agreed that for newly introduced sequences, then their safety should be assessed. For instance, the allergenic potential through in silico testing. And it was also suggested the relative quantification of recombinant DNA versus total DNA. We also discussed, of course, about omics. This is a cross-cutting issue across all the sessions. Particularly here, we discussed about the integration of omics techniques in the regulatory risk assessment of engineered microbial cell factories. And, well, the main conclusion was that currently, omics techniques other than genomics have certain limitations for their routine use in the risk assessment of GMM fermentation products due to the need for harmonization, internationally accepted standards and baselines, also the reproducibility of the outcomes, considering, for instance, the fermentor or culture effect. Also challenges in the interpretation of the results and also in the comparability of the data. However, well, let's finish with a positive message. It is expected that future developments in automation will result in reduced cost and will allow also the standardization that is needed to integrate these technologies in the regulatory risk assessment. So I think that, Hele, I would be all from my side. Thank you very much for your attention. I'm happy to reply to any questions later.
Thank you very much, Esther, for this. And then we have the last breakout group, and the rapporteur there is Gabriela Prekap.
Yes, good morning, everyone. Thank you, Hele. So in our breakout session number 4, we discussed the developmental needs for the safety assessment of food ingredients derived from precision fermentation. I would like to take this opportunity to also thank to all the stakeholders involved for the very lively discussions and for the relevant feedback, and of course to all the EFSA colleagues for the great support provided. So the first question we had was related to the potential process and product hazards associated to the precision fermentation derived products. The participants reported that the main hazards would relate to the chemical and biological hazards. So we have these examples of potential residues that might end during the process, heavy metals, pesticides, antimicrobials, growth factors as well as biological hazards referring to the potential presence of residual DNA, antimicrobial genes, a possible biological contamination with unintended microorganisms, spontaneous mutations, and referring to the product, potential toxins, secondary metabolites, also post-translation modifications, which the participants reported were more specific to the precision fermentation-derived products that could end into alteration of product integrity and of protein function, potential allergenic protein byproducts, as well as the relative similarity and dissimilarity to the native substances. So these hazards are actually considered in the risk assessment, so they are not new to us. But there are some particular hazards specific that I already mentioned. With regards to the chemical and molecular characterization and the identity of precision fermentation-derived products, the stakeholders mentioned that the level of information required could depend on the product. For instance, we had some examples in the subgroups referring to the protein, to the sequence of homology, sequence and structural similarity, including as well the post-translation modifications that could be characterized by the different techniques that you can see on the screen. When it comes to the appropriateness of the analytical methods, the participants highlighted that there is a need for further development, for instance, for the quaternary structures of protein. They were mentioned that are more difficult to assess, and the in silico tools could be an alternative approach that could be integrated to help in the assessment. When it comes to the acceptable level of identity between the precision fermentation-derived products and their native counterparts, the stakeholders highlighted that we might need a consensus between the risk assessment and the risk managers regarding the acceptable level of identity, and the comparative approach could be applicable, of course, only if a natural counterpart exists in nature. And they were also wondering how similar a similar product has to be. With regards to the fate, so digestibility, bioavailability, and ATME in the gastrointestinal tract of the precision fermentation-derived products and allergenicity. So the fate could help to unravel the real exposure, bioavailability, and ATME. And it was highlighted that the harmonization of methodology is fundamental. With regards to the allergenicity, it was generally agreed that the weight of evidence approach is sufficient for products similar to native substances. And for the novel products, it was also mentioned that the lessons could be drawn perhaps from the ICAH guidelines on allergenicity for biotechnology products, so from the pharma sector. When it comes to the fit for purpose of the toxicological approaches, it was a general discussion that the classical toxicological studies may not be needed, so they might be not fit for purpose to assess, for instance, macronutrients, such as the proteins. And it was also generally agreed that the animal testing should be reduced or not used, which is also in agreement with the EFSA strategy that focuses on the stresses also the minimization of the animal testing and also the integration of the new approach methodologies that should be integrated in the next generation assessment. And here we had the example of the omics that could be very relevant to be integrated in the risk assessment. The last questions we had were regarding the additional considerations for new-to-nature precision fermentation derived products. So they refer practically to the products that don't have a counterpart in nature, so they could be new in nature. And it was generally agreed that there could be concerns for allergenicity, as well as imbalanced nutrition by altering the bioavailability. And with regards to the post-marketing monitoring tools to check if adverse and side effects are observed, if there are any, they should be in line with the general use of the product, reassuring that the safety or if any new unforeseen adverse effect are observed. And the stakeholders also emphasized regarding this last question that they should be open and should provide as much information as possible for the consumer and to follow the rules of the full transparency. And this consumer acceptance should be also carried out to help in this matter. So these were the main issues discussed. We had more technical discussions that will be also reported in the report that we'll have after the colloquium. Thank you for your attention, and I would be happy to reply to any clarification. Thank you very much.
Thank you very much, Gabriela. Then it's time for discussion, and I would like to invite all the rapporteurs to take a seat here on the scene. And the floor is open. Please raise your hands, and let's clarify things if the reports back were not completely clear, or if you have additional comments, please feel free. There were lively discussions during the breakout groups. Do you feel that everything was covered? Please, yes. You have the floor.
Jette Young from Aarhus University, Denmark. I was wondering, when we were looking at your presentations, that all of you mentioned omics techniques in some shape or form, and some thought it was completely central, some thought it may be used at a later stage, and some thought it's completely irrelevant. So could you please comment on where do you see this? Because this was obviously something that was relevant for everything, but very different approaches.
Yes, please, Wolfgang.
Yes, indeed, we had quite some comments which differed among the four groups, but I would not say that the groups themselves, at least not for breakout session 1, that there was general agreement that omics is not useful. There were such comments, and in particular with regards to cell culture-derived foods coming from animals, there were some comments that for the consistency of the product, that may be a tool. While I think the situation is very much different for precision fermentation, genomics. I mean, it's a very well-established approach and method, and without, I could not imagine even about precision fermentation products. But first, Emma, you want to add something on the cell culture, and then we go to the precision fermentation?
Yes, sure. So I think something that was perceived was, in some cases, there is opportunity to implement, for example, new approach methodologies and omics in analyzing the products, but it's not clear if this is like necessary towards investigating the safety of this product. So there are some gray areas and lack of understanding if the implementation of these techniques would be beneficial towards establishing the safety of the cell culture derived foods and food ingredients. So there is a bit of a hesitance sometimes.
Can you hear me? Yes, I was not sure, sorry. Yes, well, as Wolfgang mentioned, the case for precision fermentation is a bit different because, as you may know, whole genome sequence is, let's say, part of, is used for the characterization and risk assessment of microorganisms according to our guidance documents for identity, characterization of genetic modification, also for potential traits of concern. I mean, genomics is fully implemented. And the discussion will basically highlight that other techniques rather than genomics might not be like so ready to be integrated. Of course, they could be extremely useful, but they are not so ready for some of the issues that I mentioned before. But, well, if I'm not mistaken, you can correct me, Gabriela, but it's also EFSA's vision, as you say, no? That by 2030, the omics NAMs will be integrated in our risk assessment. Thank you very much. Sorry.
Thank you.
Gabriela?
Maybe on the proteomics, the application of proteomics may be an issue. I mean, especially when it comes to cell culture-derived foods. One of the central key points in the risk assessment is also the comparison with the conventional counterpart. Such foods does not need to be similar or even the same or even similar, but that proteomics could be a tool to compare it. How you interpret that's another thing, but of course the more similar you are, the more you could argue that it's not so much different from the conventional counterpart.
Okay, yeah, one more question over here.
Yes, to support, because we are implied in the omics part in EFSA, so we are currently funding a roadmap where omics, different omics can be applied in the risk assessment, so covering all areas of EFSA. And within the discussion in the working group, it was, for example, about metabolomics, no? When you look what you have there in the fermenter, one time can be something, the next time can be something different, independently if we are talking about a preservation, fermentation or not. So for the moment, it's still very difficult to interpret the data that you get from the analysis and the variation, the natural variation. So there is still something to be done there. And also the costs. No? It was told that the costs are currently very high to be used in routine, but we expect that this will change in the future. Thank you.
Thank you. More comments? Yes, now I see two hands. First here and then, yeah, okay.
Thank you, Hans Verhagen, consultant. I see across the board there is a general tendency now to get rid of the classical toxicology tests, the gene tox battery, the 90-day study, etc. Is EFSA already prepared to consider and accept dossiers in the absence of these classical tests? Because they probably will not answer any questions, only spend money, time, etc.
I mean, there are different ways to demonstrate the safety of novel foods, and if you would have a look on all the way back, there are some novel foods where 90-day studies were not requested, and a few also not even genotoxicity tests. The question is always how. It relates always on the type of products. For some products, new components, new substances, extracts from plants where you can only characterize a fraction of the molecules and compounds which are inside. At the moment, I think we're not there where we can say we can abandon 90-day studies. So I think for some type of novel foods, it's still one of the core evidence that needs to be provided. Yeah? For whole foods, where we know we have limitations to test whole foods, where you cannot feed 200 times the higher dose to rats than what human may consume. There's an inherent limitation there. There's a protocol to address this, but also limitations are there. And on such type of products, and I think that from the discussion what we had and from the knowledge we have gathered so far is, at least for the cell culture-derived foods, our approach is a tiered approach. So first, we always start with the production process. We need to know exactly what is in, so that nothing can be hidden there. We not necessarily need to publish everything, and we should not with regards to very innovative issues, as long as they do not concern the safety of the final product. But the idea is to have a tiered approach, go very thoroughly through the production process to see what is in the final product, do a risk assessment of the components of concerns, and then, if possible, to avoid a 90-day study. The versed documentation is if there are uncertainties with regards to components used in the production process, new components with an unknown toxicological profile, the more likely it will become that a 90-day study may be requested or will be requested. But I think, in theory, for cell culture-derived food products, the idea would be to avoid 90-day studies. That's also in line with the novel food regulation. If such tests can be avoided, they should be avoided. With regards to new technologies, EFSA is hiring people. They are working. But I must admit it's very difficult, and validation is the key, validation of new methods before they can replace the classical toxicological tests. Yeah. It will come, but I'm afraid we're not there where we would like to be at the moment.
And it's the Park project is also working on this, on the research side. Thank you. I think it was a question down in the middle of the... Yeah.
Yeah, hi, Valeria Telori, Mozambique. I would like to expand a bit on the note you had about growth factors as processing aids if they are not found in the final product, and what would be your assessment when these growth factors are not found in the final product, and these, as you mentioned, for insulin, for example, can be recombinant if they are used as processing aid and therefore are outside of the scope of looking for residual DNA, because as we move away for pharma-grade components and the requirements for food-grade growth factors are not yet defined, what will be your approach to assess the necessity for residual DNA testing for recombinant growth factors and protein if they are not found in the final product? Thank you.
Yeah, I think it's back to Wolfgang.
Okay, there are, I think, two questions. One refers to growth factors in general, and the other one I understood refers to residual DNA. I understand from the legislation if recombinant DNA is found in the final product, then it cannot be a novel food, but then it would fall under the GM regulation. Yeah? I'm not about 100% sure what about, because I also got this question during break, what about if you're using a recombinant protein where the recombinant DNA can be found in the recombinant protein product, which is then used in the production, but then in the final product, because of the dilution and the washing out, you cannot detect it anymore. This I don't know whether such type of products would still fall under the GM, but I don't think so. But I'm not totally confident. Maybe somebody knows from the Commission.
Yeah, please, over here.
Thanks. I think we have to distinguish here whether this protein or growth factor, which is added to the meat, is marketed previously from a third-party producer or not. In case of yes, so it's not produced in-house, but you purchase it from the market, it will be the producer of a growth factor who has to comply with the GMO legislation if it's case. And if there is recombinant DNA remain in the product, it will be considered a GMO, it will have a appropriate level and everything, but it doesn't mean that the final meat will be considered GMO. The ingredient, let's say, it will comply with the legislation, with the permission, with the labeling support, and this case is the easy one, I would say. If this growth factor, hormone, protein, or whatever is produced in-house and added to the manufacturing process, then it's the final product has to be tested. And my understanding is the same as yours. Depending on whether DNA can be detected in the final product, it could be in the GMO legislation or out of it. But it will have the final product to be tested according to the standards for finding recombinant DNA in the product. We will tell if it's on one side of the legislation or on the other one. That's my understanding on the situation.
Colleagues from the European Commission, if I may ask you, you have a view on that? And you don't. It's not a binding thing. Yeah, nobody can make you liable for what you're going to say.
No.
No.
Yeah, I'm from the GMO unit actually. So I would say it's the same as you said. So if you test the final product and you indeed find recombinant DNA there, it's in the GMO legislation. If you cannot find anything, then you are in the novel food legislation.
Thank you. Are there more questions or comments?
There was a second question there, because what growth factor? I think
Okay,
it was independent
yeah,
even
sorry.
from the DNA, if I understand correctly. Or was it related everything to DNA? Okay.
Please wait for the microphone. Sorry.
Does this apply to compounds that are used as processing aids, so not as ingredient of the food? So I understand all these comments for ingredients, but not for processing aids.
Can I comment? Can you hear me?
Yes.
So we had this discussion about processing aids in.
I think it's working. It is working. Okay.
So we had the discussion about processing aids. What could be characterized as a processing aid in the production of these foods? And one thing would be if something is expected to be in considerable concentrations in the final product, and another parameter to be considered if there are a priori safety concern regarding this specific component. So it's like a combination of parameters to be considered towards, let's say, deciding if something would be considered a processing aid or not. But yes, growth factors is one example, but maybe there are some other components used that could be also investigated under this prism.
Yes, one more in the back of the rows.
Hi. Is it on? Yep. My name is Christian Kalk. I work with Meetable as Regulatory Affairs Manager, and I'm also an independent consultant. I'm just thinking loudly here when we talk about growth factors. Can't we draw, for risk assessment and for formal approach, draw a parallel to the way we are dealing with food enzymes that nowadays also typically come from GM microorganisms?
Wolfgang, do you want to
I would
comment
say
on
yes,
this?
yeah.
Yes. That was the short answer.
Actually, Christian, this was in the feedback we received from the EU survey before this colloquium. It was one of the proposals from the registrants that this could be dealt in a similar way with the food enzymes, and then all the things that the colleagues mentioned before would be probably applicable to establish the safety of these components.
Anna, please.
Can I continue on that topic? Yes was the short answer, but what does that mean in practice? That we would have a pre-approval procedure for those particular components, and then we would have like a list of approved growth factors, processing aids?
I understood it was related to the DNA residual, so that, yeah, the question. Yeah.
It's also a comment over here.
So we are crossing answers. So can I ask you the question again? Because it was about food enzymes and having growth factors being assessed, processed in our system, the same way that we're doing for food enzymes. And my question was, how would you see that happening? So sort of like, first we submit all the growth factors, and EFSA goes and works on it, creates a list, and then we do it, or?
I think there is no legislation in place for growth factors, so that does not require a pre-authorization as a growth factor. But I think the question was entirely related to residual DNA. Yeah.
Mm-hmm. Mm-hmm. Yeah. You wanted to make another comment? Okay. More questions? Comments? Are you eager for coffee? Seems so. Then I would like to direct a warm thank to all the people who have been working on summarizing the outcome from these 4 hours of discussions in the breakout groups. It's 4 hours in 10 minutes each. It was you have been doing this during the evening and this morning, so thanks a lot.